honored to introduce today's speaker, Professor Cheng. I'm a postdoc in his lab. Dr. earned his bachelor degree in fluid mechanics at Zhejiang University in China, and a Ph.D. degree in bioengineering at Columbia University in 1988. His Ph.D. and the postdoc work with Dr. Richard Scott was on mathematical modeling of cell locomotion and cell adhesion. But now he has converted from a theoretician to a experimentalist, pioneering two-dimensional analysis of cell adhesion and signaling molecules. Dr. joined Georgia Tech in 1990 as an assistant professor in the Department of Mechanical Engineering, and went on to become associate professor in 1996 and a full professor in 2001. He's currently Regents Professor of Georgia Tech and G. Erskine Love Endowed Chair in Engineering. Dr. has earned many numerous awards, including YC Fong, Young Investigator Award, and Presidential Faculty Fellow Award, and Hemorrheology and Microcirculation Award. He's also an elected fellow of both American Institute for Medical and Biological Engineering and Biomedical Engineering Society. Now, without further ado, let's welcome Dr. Cheng to tell us about mechanoreception of adhesion signaling molecules on the cell surface.
Thank you, Bao. That was a long introduction. I think people know a lot of this already. Bao, you alluded to that I've been here 25 years, and in 1990 came to Georgia Tech, and Georgia Tech is not what it is now today. Very different. So I gave that title to when Bob, Cody asked me to do this Breakfast Club. I gave that title. I didn't know what I was going to talk about. I gave a title anyway. Then yesterday, when I was trying to put together slides, I thought that I stopped. My talk was largely on the molecular or mechanobiology at the molecular scale. So this is maybe a more appropriate title, and the reason for that is really it links to another title that why does it not change? Here is supposed to be cellular scale, and for some reason I did not change my slide. Anyway, so this is when I came to Georgia Tech. I couldn't find the original image that Dr. Robert Nerum, who the former director, the founding director of this Petit Institute, published. So I got somebody else's image from the internet. But this is what I saw when I came to Georgia Tech, when I was trained as a theoretician and then want to become an experimentalist. And I look at, this is, I guess, very well known. It's when you have endothelial cells cultured in the static or oscillatory condition, the endothelial cells are this cobblestone shape and rounded and with no alignment. When you apply a steady state shear stress flow over 24 hours, you will see the alignment of the endothelial cells in the direction of flow and elongation as well. So that was in the, I think, Dr. Larry McIntyre, very familiar with that because he also contributed a great deal to this back when he was at Rice. So, and that's molecular, that's mechanobiology at the cellular scale. So, and fast forward to today, I want to start with some simple questions and then progressively make it more and more specific and more maybe harder to do. So if you want to demonstrate mechanical force triggered cell signaling, the previous image and many work follow that already tells us that this is correct. So the hypothesis has been validated because you expose the cell with wall shear stress or many different forms of mechanical stimuli, you will see the response. And that response was due to signaling. Okay? But then, now I'm going to have the same thing here, mechanical force triggers cell signaling, but inserted here a phrase saying that mechanical force exerted on the cell surface receptor via engaged ligand trigger signaling. So that will be more specific, and the question will be, what is the cell surface receptor? Right? Now, do it a little bit more specific again, and I'm going to say that mechanical force exerted on the cell surface receptor via engaged ligand, it regulates signaling initiation in the cell. So here, you could have force applied to the cell surface receptor. And then the signal initiation may or may not be occurring at this cell surface receptor, right? So how do we figure out whether or not this cell surface receptor really convert the mechanical cues into a biochemical signal? We started looking at the signal initiate by this receptor, and we want to see the mechanical force, whether or not it does anything or regulate this signaling initiation. Now, I would present data to here and try to, I think the data here would support what we can do, but down here it's a little bit fuzzy. So I put down here mechanical force exerted on the cell surface receptor via engaged ligand, regulate signaling initiation in the cell via mechanism. Don't know what it is yet. All right. So let me start with a set of questions. So we ask, what are the mechano sensing molecules on the platelet? I can pick a particular cell. In this case, it's platelet. And then here, what is a mechanosensing molecule? And it looks like these two questions are quite similar. One is plural, one is singular, but no, not really. So if you ask the top question here, the answer will be, well, provide a set of possible or candidate molecules, right? So on the cell surface, the glycoprotein 1b95 complex, which we'll focus on a lot. Integrin is another molecule we'll focus on today's talk. Yeah, we'll work on T-cell receptor, a lot of the other things. And then you can look at molecules inside the cell, cytoplasmic, cytosolic molecules like vinculonectin, a lot of them. And extracellular, I'm going to start out by talking about von Willebrand factor. And I know a lot of people, like in this campus, I can think of Andres Garcia and Tom Bacher was very interested in fibronectin. Right? So then, what is a mechanosensing molecule? It's the definition of a mechanosensing molecule. So it's not a possible molecule that may do that. Really trying to kind of dissect different aspects, or if a molecule plays some role, you look at any of those candidate molecules, what exactly their roles are. I break it down into mechanopresenter, mechanotransmitter, mechanoreceptor, mechanosensor, mechanoresponsor. Now, the title of my talk is really down here, but I'll touch on the other things as well. So here's definition. So a mechanopresenter is a molecule that presents information to a mechanoreceptor upon change in its mechanical environment. A mechanoreceptor is a receptor that receives the mechanical cues via engaged ligand. A mechanotransmitter is a structural molecule that transmits the mechanically encoded information over a distance. A mechanosensor is a molecule that converts the mechanical signal to chemical signal at that molecule or a assembly of molecules. And then a mechanoresponder is a molecule that changes its mechanical behavior in response to intracellular signal. Now that I think about it, maybe I would not necessarily say intracellular signal. Well, let's think about that. All right, so I want to start it out saying, give you a first example, and this is the von Willebrand factor, or VWF, as a candidate mechanoresponder or a mechanoreceptor. Okay, so this is one of the application area of my lab, which is hemostasis and thrombosis. I'm showing here it's a disrupted blood vessel. Here are the endothelial cells, and the disrupted blood vessel here expose the subendothelial matrix. And then the platelet here are interacting with the von Willebrand factor here via the surface molecule GP1B-alpha, or a integrin alpha 2B beta 3. There were other molecules there, but let's focus on the von Willebrand factor. So this part of the work was done by a postdoc, Zhenhai Li, somewhere here. All right, Zhenhai, thank you. So down here is the schematic domain organization of a von Willebrand factor monomer. In the blood plasma and also immobilized on the subendothelial surface, von Willebrand factor actually was presented as multimers, very, very large multimers. So the domain that interact with the GP1B alpha is it's a domain here, A1. That's three domains together, very similar, A1, A2, A3, but A1 is the domain that interact with the GP1B alpha. Now, I'm going to introduce another protein called ADAMTS13. It's an enzyme in the plasma. Its role is to cleave the multimeric von Willebrand factor into smaller pieces, because the larger the von Willebrand factor, the higher the adhesive activity. All right, so you don't want your VWF to be too big, and there are diseases that you have ultra-large von Willebrand factor that not cleaved by this enzyme, and disease called TTP. So this ADAMTS13 enzyme will cleave at the A2 domain right there. Now, it's well known that the cleavage of the von Willebrand factor by ADAMTS13 occurs very efficiently in the bloodstream, not at all in a static condition. So this important function that regulates the adhesiveness of von Willebrand factor in your bloodstream is regulated mechanically. All right, so now we're going to look at how fluid shear stress affects the cleavage of this. So we have two constructs, and breaking down this molecule, take a piece here, it's called a TSP7, a fragment of this molecule, and then another fragment of that molecule called a cup. All right, so if you take ultra-large von Willebrand factor, you can obtain that by stimulate your endothelial cell with histamine. You look at it under confocal microscope. This is 10 micron bar, and it looks something like that, fluorescently labeled. Now, you've applied a shear flow over it, and you immobilize this with a high density of antibody against von Willebrand factor, but low concentration of VWF, so that there were a lot of antibodies on the surface. Only a small number of them are used to capture the von Willebrand factor. When you apply shear flow, these things get stretched out and then bind to the available capture antibody so that when you stop the flow, you look at it under the microscope, you can still see it stretched out. So this is what you see. Flow is going this way, right? So you can think that a molecule is mechanically responsive because applied force, it changes its conformation, right? So we use the atomic force microscope to study it, its interaction with atom TS13 or its construct. So this is a AFM that we use, and you allow the two molecules to contact. You can either, then you put it out, you either not observe any interaction, or you observe interaction, in which case there was a spike here showing force. Now, or you can do it in the other way, which is you hold it at a constant force and then look at how long it lasts. So that's the rupture force measurement in the first case, and this is the bound lifetime under force measurement. All right. So, so if you can detect whether or not binding or not binding is a binary readout, you can then look at the frequency or the likelihood of whether or not binding occurs. So it's plotted here. You do 100 of touches and count how many of those touches resulted in binding, and that's what is frequency here. So if you look at BSA as a control, it doesn't bind to either the full-length atom TS13 or its fragment TSP7 or cup. But now if you change it to VWF, it binds to atom TS13 well, and the cup domain well, but not TSP7. So you would say, aha, the atom TS13 binds to VWF via cup domain. That's a binding site that you can identify. Now that's when you do the experiment in the absence of flow. Okay, so it's a kind of a globular image that you remember in the previous slide. Right now, there was some calcium dependence because you can use EDTA to chelate divalent cani, and then the binding was suppressed. This thing doesn't do anything, so that's just the background. Now, look at this part here. So if you add cup domain to the solution of your AFM experiment, that would block the binding of the binding site on the VWF. So then atom TS13 on the surface would not be able to bind, and that's what its reduction here, right? Also the reduction here, the cup domain in the solution blocks binding. So, but if you use TSP7 in the solution and you put it here, that does not block, right? So that's all making sense. If you measure the bound lifetime as a function of force, atom TS13 full-length molecule and the cup domain give you same kind of dissociation characteristics. Again, saying that atom TS13 bind to VWF, the globular one, before you apply any forces, with the cup domain. Okay? That's what it stays here in a divalent cation dependent fashion. So now, this is binding of atom TS13 contract to VWF after you shear it. And you saw the image, that was the stretched out condition. So BSA control, no binding, and this is static condition at the high coating concentration, no binding, but then if you dose responsively increasing the coating of the VWF, you see the binding frequency progressively increase until you really saturate it. You have too many VWF that not enough antibody to capture them when they stretch out. So then they recoil back to the globular conformation in this case. Okay. Really, the cup domain doesn't do anything, except that in the very, very high concentration, they are globular, then you see it binding. Okay. Now, if you then measure the bound lifetime under force, and you will find the atom TS13 curve and the TSP7 curve behave very similarly, right? So now, this data would say atom TS13 bind to stretched VWF via P7 domain. All right, so let's see what we can conclude in this example. So VWF is a mechanoresponder because it changes conformation in response to the fluid shear stress. VWF is a mechanopresenter because it presents different binding sites to different domains of atom tier 13. Right? So I hope this example, without solving yet, illustrate the concept of mechanoresponder and mechanopresenter. Now I'm going to move on to talk about a little bit what is the big deal, what's the physiological relevance. So this is a cartoon, got it to this website. Okay, Zhen Hai got it for me. So the von Willebrand factor was actually made largely by endothelial cells, and under inflammatory condition, it secreted a very large size or ultra large von Willebrand factor, and it's reduced in size by atom tier 13. If you don't do that, you're going to get TTP, thrombotic thrombocytopenia purpura. It's a disease that one form of it, I think majority of them are actually autoimmune disease where the patient developed autoantibody that block the activity of atom tier 13, so we could not cleave this. And as a result, you have platelet aggregation and formation of microthrombi. So it is a physiologically important question. Now I'm going to move back to this definition here, and then I'm going to say that show two other things. So receptor and mechanosensor. So give the second example is GP1B alpha as a candidate mechanoreceptor and mechanosensor. Later on, I'm going to replace this of an integrin alpha 2 beta 3 and say that is another candidate for mechanoreceptor and mechanosensor. So go back to this cartoon that shows the thrombotic and hemostatic cascade where platelet adhere to disrupted endothelium. So the work I will present is done by two graduate students. Arnold G. graduated, I think, December last year. Yunfeng Chen is still in my lab. Are you here, Yunfeng? He doesn't have to listen to that. All right, so let's go back. So we mimic the, whoops, that's, how can I go back to where I wanted to? All right, so we mimic the platelet translocation on the endothelial, subendothelial matrix using a device called Biomembrane force probe. So you grab a platelet with a pipette, and then you have a red cell serve as a mechanosensor. It has a bead here, and then you do the measurement. So this is a von Willebrand factor A1 domain, and these are a GP1B alpha. And inside the platelet, we can also look at signaling. Okay, so that's the real image of a biomembrane force probe. It's a platelet here. You had the molecule of interest. So here is the A1 domain of von Willebrand factor, and here are the GP1B 9-5 complex. The 1B alpha is this long guy. And there were also integrins here that I will touch on later. You see this cartoon, and that's the real video of how the experiment were done. You can look at things here. So this is a rupture force. You get a force when the retraction. Here is the contact, right? So this is a bond lifetime measure at about 20 picomillion force. Okay, the same thing that you've seen before. Now, in addition to that, we add another optical path so that you can see the on the fluorescence channel intracellular calcium imaged by a calcium dye. So then you would get, this is the calcium signal here on the left ordinate, and here is each of these little triangle is a interaction that you see in the other video channel for the binding, and then how long the bond lasts, it's kind of here. So this is the cumulative lifetime. Okay, so a few control experiment. If you just aspirate the platelet, a platelet is very, very easy to get activated. But we get good at it now. Or, Arnold and Yunfeng get very good at it, so you don't generate any calcium by just aspirate the platelet. Or you can use a ligand-free beads to touch it repeatedly. It doesn't really generate any calcium until you put the ligand of von Willebrand factor A1 domain there. Then you see lifetimes and then also calcium, right? So I'm going to start conclude that the intraplatelet calcium is triggered by GP1B alpha mechanoreception rather than non-specific mechanotransmission. Because aspiration pressure and impingement of the pipette, of the platelet, did not generate any calcium signal. Right? So now you've seen the images here and the data graph here. This is interaction with a wild-type von Willebrand factor A1 domain. There were bleeding disorders called the von Willebrand disease with mutations in von Willebrand factor. Patients with those diseases has a longer bleeding time. And if you use a mutant von Willebrand factor A1 domain that was found in patients with the bleeding disorder von Willebrand disease, and you see different kind of signals. And so here is the bound lifetime versus force, and this is the data actually from a 2008 paper that that's my first collaboration with Dr. McIntyre. He is the one that got me into study of platelet. So now then the bound has a different force-dependent lifetime characteristics because with the increasing force, the bound dissociate faster and faster, or the lifetime is shortened by force. And whereas in here, the lifetime is first prolonged by force before it shortened by force. So we call this catch bound and this slip bound. So is this interaction characteristics between a von Willebrand factor, either wild type or mutant, that you use to exert force on the platelet, GP1B alpha. You had a different way of exerting force, so there were information encoded in the way that the platelet sees the force. With the platelet able to interpret that information and respond accordingly. So here is the calcium signal. We take the peak value as a single number to reduce the representation of data. So each point, it's a platelet that tested 5 minutes. And this axis here corresponds to the bar, and that's the bound lifetime. You saw that plot. So it was 10, 25, 40, 60 piconewton forces, and it's short lifetime and then prolonged by increasing force and then shorten again. All right, you see the two patterns matches pretty well. Right? Now, if you use a mutant that is a von Willebrand disease mutation, you see this is the slip-bound pattern on the bars. The calcium signal also followed the same trend. You can even use an antibody against the GP1B alpha that gives you a slip bound, and the signal follows that. Now, if I change the way how force is applied, instead of applying a durable force, meaning that you apply the force to a level that you select and then hold it there until dissociation occurs, in that case, the longer the lifetime or the longer the duration of the force, then you get a higher calcium signal. Now, if I just run the force until rupture without holding it, so that's the rupture force experiment. What you see here is this data. Same thing, calcium here. So this is the rupture force. It's actually much, much higher. You see this one, it's 126-ish piconewton. These number here is how fast you ramp it. And it's well known the faster you ramp it, the higher the rupture force is. Right? So that's the basis of dynamic force spectroscopy. And this corresponds to the same molecular interaction. And you see no calcium generated if you do it that way. So the force has to be sustained. This is the information encoded in the mechanical force pattern that was responded or interpreted by the platelet and responded with a calcium signal pattern that matches the bound lifetime but not the force. Okay. So what you saw before are kind of pool data, sample analysis. You measure many, many bound lifetimes. You can average and etc. You can also look at a single platelet, and each platelet gives you a calcium value, the peak calcium value, and then you look at the repeated contact and dissociation touches and measurements of the kinetic on that particular platelet, and just look at a longest lifetime prior to the calcium onset. Right? So there were several interactions occur. You don't see any calcium, but then boom, calcium come up. What happened? Why wouldn't it start in the beginning? Well, some of the earlier ones are short. So if you just look at the pre-calcium longest lifetime and this axis and you plot the calcium level there, you see a pretty good correlation. And same here, a different level of force, even for the wild type and for the mutant. They all kind of group in that. All right? Another measure is how fast calcium come up once you had this longest lifetime. So this is the pre-calcium longest lifetime. Here is the delay time from when you see this longest lifetime event to the time when the calcium spike comes up. So, of course, the stronger the simulation as represented by the a prolonged lifetime here, the shorter you have to wait. Right? And you can actually look at whether or not this kind of determination, so there was a, you can set two thresholds, and this is below a threshold, this is above the threshold here, and this is below the threshold and above the threshold. I would say if the bound lifetime is above the threshold, whether or not you would see a calcium. When it's below threshold, whether or not you see a calcium. So when you have a lifetime that was above the threshold here, you would have calcium. So this 38 events that satisfy this criteria. Now, when you do not have a long lifetime or below the threshold, which is in down in that axis here, and then you also do not, here, and you also do not observe a bound lifetime. So it's, again, these two bars saying that this criteria work well. Now, would this criteria, if you use it, would you generate false positive or false negative? Yeah, you generate 1 false positive and 5 false negative. So the criteria is pretty good. Okay, so let me conclude. The second conclusion here is that GP1B alpha is a mechanoreceptor because platelet signaling is triggered by force it receives. GP1B alpha is likely a mechanosensor because it can interpret information encoded in the force waveform. Okay, so let me move to the next, the other molecule on the platelet, which is integrin alpha 2B-beta-3. So you've seen this biomembrane force flow setup. Now, we want to look at the interplay between this molecule here and that molecule there. We isolate the interaction here first. Now we want to see whether or not the signaling induced by the mechanoreception of this molecule leads to a activation of that molecule. So it's driven, it's shown in the banded conformation, indicating the integrin is in the resting state. It would not bind to ligand. until it gets activated. So, and this is a simpler just experiment. You count 1, 0, right? So we don't even measure Brown lifetime. So if you do a repeat of, let's say, 200 times, and you count the positive ones and also count the negative ones on the bottom, there's too many to count. You do the running frequency, and here's BSA control. It's very low, very clean. But then if you added a ligand, but then control how long the contact lasts, 5 seconds versus 10 seconds, well, I need to change this to 0.5 and 1. You see the adhesion frequency changes. So that's the basis of using this assay to measure interaction kinetics. That's done by Scott Chesler a long time ago, published in 1998, my first student, I think. He looks old because he is now. I couldn't find a younger picture of him. So if you plot the y-axis, the adhesion frequency, which is the average of multiple contacts and the positive event of multiple contacts, versus the contact time, you see a curve like that, right? As you increase the contact time, the adhesion frequency increase and then levels out. And you can now, and these are the two students that did the work. Now, if you use a biomolecular interaction model, receptor plus a ligand forms a bond, and that's the probabilistic version of the kinetic equation that we typically use to describe it, and then you fit it. First, there are two parameters there. It's the molecule, how many molecules on the platelet surface, on the bead surface, you can measure them. And then you fit it with this equation, that's the curve fit, and then you generate two things. One is the affinity, the other is off rate. So you can do this calculation. Now, so remember we're on the platelet. So that's, you actually have two molecules, right? So if you use ADP to activate the platelet so that the integrin become in a high affinity state, capable of binding ligand, and you present the platelet with both the reversible factor that binds to the GP1B alpha and also fibronectin that binds to the integrin alpha 2 beta 3. You get a curve actually look like that. So it's a bimolecular interaction. You get two receptors and two ligands and form two bonds, right? And the equation that, so here you actually have bond 1 and bond 2, right? That's only when the two molecular interactions are independent. They are not, right? So if you really look at the real data, it actually going like that. So what about these data and that data? So if you block the integrin interaction using either RGD or use the anti-alpha 2 beta 3 or Integrin that would block the binding of the integrin, you get this curve. If you do not block them and you use a nice resting platelet, initially in the short contact, you see the curve follows pretty much like that. But then as you get longer and longer contact time, it actually matches this. This is without activation by any agonist, right? It's a activation by the engagement of the first receptor-ligand pair, and then activate the platelet that leading to the activation of the second receptor that binds with the high affinity. Okay. So let's see. All right. This experiment wasn't a most desirable one because it's concurrent presentation to the platelet of multiple ligands, and the platelet activate during the process, right? So the two students, Yunfeng Chen and Arnold G, devised a what's called a switch bit experiment, where we separate the presentation to platelet multiple ligands in space and time. And this is a later version done by a new grad student, second year. Her first project is to get another pipette here. So now you get two BFP here, one pipette here to grab a platelet. So this bead's coated with one ligand. That bead coats another ligand. So one reversible factor A1, fibronectin here. The platelet touches this bead first, and then you see it no calcium, and then after you get calcium, you move to the other side to touch the other one. So the presentation of the ligand to the platelet is now separated in space and time, right? So here's, again, the data from these two students. If you had a discoid platelet that you presented to fibronectin, before you tickle it with the von Willebrand factor A1 domain, they don't really bind well. Go back here. They don't really bind well. Now, if you activate the platelet with ADP, then it binds, the alpha 2 beta 3 binds to the fibronectin very well. So these are the kind of negative control and positive control. If you do the switch experiment, you interact the platelet with von Willebrand factor A1 domain, you observe the calcium triggering, and then you switch to touch it with the fibronectin coated beads, you see occur something like that. It's in between the two. So this is a integrin alpha 2b beta 3 intermediate affinity state that has been identified by this speech assay. You can block it, so it's definitely mediated by the beta 3 integrin. This is a specific antibody for beta 3 integrin. So you can calculate the affinity this way. So this is the low affinity state, high affinity state. These two are intermediate affinity state. Okay, I'm not going to talk about this. It has to do with signaling. So you can also use conformation reporter antibody. That a lot of them are available for integrins, and there were 3 antibody. One binds to here. It will not bind a bent integrin because the binding site was buried. So this antibody report the extended conformation of the integrin, and the other antibodies report the swing out of the hyperdomain, and this hyperdomain swing out signifies the high affinity conformation, and this is another antibody that binds to the active binding site. So if you use ADP to activate the platelet, all 3 antibodies will bind. They are all binding sites are available. If you have a discoid platelet, none of the antibody will bind. That suggests that it's in this conformation. If you use the von Willebrand factor A1 to engage the GP1B alpha, what you see is that only this antibody binds, suggesting that the hyperdomain has not swing out, and the active headpiece here is in the resting state. Headpiece is the resting state. So we can identify a conformation of the integrin that corresponds to the intermediate affinity that you measure. So function, structure can relate to that. Now, so remember we're doing this repeat test of 200 touches and look at the running frequency. The running frequency actually changes over time in this case. If you have the extracellular calcium in the medium, in the first 50 touches, the adhesion frequency estimated from these 50 touches is about the same. But then it progressively increases as you touch the platelet more and more. Now, if you chelate extracellular calcium using EGTA, what happened is actually drop. Okay? So if you compare, these are the high, intermediate, and the low affinity state. If you chelate extracellular calcium, it actually returns in 5 minutes to the low affinity state. If you allow extracellular calcium and to enter the cell, I guess you will see the high affinity state here. So looking at the last 50, that's what you'll get. This is the last 50, that's what you get. So the platelet is being tickled now by the fibronectin, which binds to the integrin alpha 2 beta 3, nothing else. And that force exerted on the integrin leads to the activation of the integrin itself. And that activation requires extracellular calcium. We did not go measure the calcium, we'll do that. My guess is that it's a outside-in signaling by the integrin when it's engaged to the fibronectin and force exerted on it, which then allow extracellular calcium to come in, which then activate the integrin, which then enable it to bind with high affinity. So you can look at the bar graph here. This is the discoid platelet correspond to the green. The two orange ones are the intermediate affinity state, either this way or that way, looking at the first 50 touches. But then this one is the last 50 touches of this guy here. This one is the last 50 touches of this guy here. They decrease and increase. And this is the positive control using ADP activity 1. So I guess the integrin alpha 2B-beta-3 is a mechanoreceptor because pulling via a engaged ligand or antibody-induced outside-in signaling manifests as a calcium influx and integrin activation to a high affinity state. So this is the conclusion number 3. Integrin alpha 2B-beta-3 is a mechanoreceptor. I already talked about that. And the question of whether or not integrin alpha 2B-beta-3 is a mechanosensor can be analyzed by the similar experiment used to study GP2B3A. So I think I'm going to stop here, not to go on to the last piece, but just saying that we can think of put all of these together into a model where you already seen this before. We use this experiment to mimic the platelet rolling, and you get the platelet from a discoid resting state to a intermediate activated state to a highly activated state, during which integrin would get induced conformational change, and then going to the high affinity state after you receive intracellular calcium. So I think I'd rather not to go into new thing, but give some time to questions. I want to wrap it up by saying that several examples have been provided to illustrate mechanobiology at the molecular scale. These examples were used to explain various roles a molecule can play in mechanotransduction process. Biomechanical analysis of molecular interaction and conformational change is key to elucidating these roles. I'd like to close by acknowledging the collaborators. We have more than this list here. These are the ones that collaborated on the data that I presented. And I already show you along the way the people who did the work. So thank them, and of course NIH support. In the end, of course, there were a lot of people in the lab in the past 25 years. This is a small sample of those, and I'd like to thank you for your attention. Matt.
Yeah. Beautiful work. I'm curious if you have any sense, or anybody has any sense of how the membrane itself affects mechanoreception. So lipid content, whether or not two receptors are in the same lipid raft, or anything like that, any studies in that.
So the answer is yes, yes, and yes.
Yeah.
So yes, membrane raft, or the organization of the membrane, affects mechanoreceptor and mechanosensing. Yes, we have worked on that, and I didn't present the data, but this is one of the two big projects in the lab. One was on the viral infection of T cell. The other is T cells infiltrated in tumor, that they behave differently in the mechanosensing, and we have data to suggest that it's due to the membrane raft structure, because you can perturb that to change it.
Hey, Cheng. So again, beautiful work. I mean, the studies you've done to elucidate these mechanisms is just amazing. My question is, when you go into the in vivo setting, how do you determine, or how can we verify which of these mechanisms are primary or prioritized during particular processes? Are there experiments that you're doing or envision to be able to do that?
Yeah. So this is really a very important question, right? So it doesn't really matter how beautiful it looks. What is important is whether or not this is play a role in physiology. We, of course, in the process of doing it, and we haven't had all the answers. So one of the approach that we'll do is to correlate the finding in these kind of in vitro experiment or ex vivo experiment with studies with, let's say, a mouse model that alter this molecule. So we are in the process of doing that, and I'm going to need your help to get animal facility space for me. We just started
That's
this.
above my pay grade.
Yeah, this is important. Great. Matt again.
Yeah. Is there any chance that you could couple mechanoreception on the extracellular surface to an alternate signaling pathway on the intracellular surface?
So
You could control another signaling pathway.
yeah,
mechanically.
I think a lot of people actually was trying to do that. I can think of Tom Barker's lab. You know, mechanical force modulate the extracellular matrix. In his case, it's fibronectin and the fibers that make up of fibronectin. And in cystic fibrosis or many other diseases. The alter the property of the matrix and alter its response to force or the mechanical environment, then the cell that normally would see or feel those mechanical cues, they no longer feel that. That leads to disease. And I think we haven't done much along that line, but it's an active research area. Von Willebrand factor is an example that we use to illustrate that. Bob again.
Yeah. So, this is great for understanding disease. A flip side would be to exploit what you're learning for example, in osteoporosis, as we age, one of the mechanisms we believe is going on is that the cells are losing their ability to sense their mechanical environment. Are there ways you can think of that we can exploit this understanding?
So that would be a goal. We haven't get around doing that yet. The lab, accumulate expertise over time, and we make progress stepwise. So, 10 years ago, we're not doing live platelets. We're doing purified proteins, and now we're doing platelet. Now we are moving into the animal model. So, Georgia Tech is an amazing place, and IBB is a good place for people to develop career. And I thought I had a lot to thank this place that I can develop a career that started out knowing nothing about experiment to now enjoy watching students doing experiments. All right. Thank you.